And it's great to see a lot of familiar faces and new ones here. Mike and I were just chatting hello on the chat box there. So, but Mike, unfortunately, that means you've probably seen some of these slides before, so you just have to bear with me on this. These are my disclosures. And again, I apologize. I was a little bit late because I just got back from clinics, which is also why I'm still wearing scrubs, because I like to wear pajamas when I work. This is hopefully will be an overview of not only just genetics and genes, but also a little bit about breast cancer, the treatments, and how we think about traditional types of therapies and breast cancers. But what really we're trying to do for both female and male breast cancer is more along the lines of precision oncology, including how we look at DNA from both the standpoint of what we inherit from our parents, but also what's in the tumor. And this is particularly important for male breast cancers, because I'm sure many, if not all of you know that if you have breast cancer as a man, you really need to get tested for things like BRCA1, BRCA2. And if that hasn't happened, then please let me know, because we'll fix that. Key to all this, though, is now we're being inundated with all this information, new knowledge, and many of my colleagues across the globe, really, they're not trained in this. And so I think it's equally important when one gets these types of genetic tests for male, and again, even other breast and other cancers, to make sure that someone who has some expertise and some knowledge in how to interpret these tests is really looking over these and adjudicating results. All right, so probably this audience already knows there's about 250,000 new cases, a little bit more nowadays, of breast cancer diagnosed each year. Male breast cancer is about 1% of that. There's about 40,000 deaths each year in the United States. And what those numbers really tell you is that the majority of breast cancers, if caught early, will in fact be cured. But 40,000 deaths is still a huge number. And again, this is just the United States every year. And so clearly we have a lot more work to do. Male breast cancer usually is what we call ductal, as is female. Lobular breast cancer is about 10% of breast cancers in general. So if you take the 1%, that means that it's even lower in male breast cancers. Other types exist, but these two really make up the large majority of breast cancer subtypes. And within those subtypes, we actually have receptors that are expressed or not. And that's how we really lump categories of breast cancer. No pun intended. I just used that word lump. I just realized that. But estrogen receptor, I think most people know about. Progesterone receptor, which is really kind of the secondary hormone receptor. If one or both or any of these are positive, we call it hormone receptor positive breast cancers. And then HER2 is a third receptor that is a little bit different. It lives on the outside of the cell, and that's really about 15 to 20% of breast cancers, and it's something called the EGFR family. The most common type of breast cancer is hormone receptor positive, HER2 negative, and male breast cancer is almost always hormone receptor positive. Sometimes, rarely, it can also be HER2 positive. I've seen that in some male breast cancers, but usually it's hormone receptor positive, HER2 negative. What we barely see, although it does happen, but very rarely, is triple negative breast cancers, which some of you may have heard about, and those are breast cancers that don't express any of these three receptors. As I mentioned earlier, most breast cancers are cured, and that's because if they're caught early, the disease is curable. And what we have to do is take out the breast cancer, look at the lymph nodes and take them out of their disease, give additional types of therapy that really are trying to eradicate micrometastatic disease. That is, cancer cells that could be left behind, but haven't yet had the time to grow and metastasize to true clinical, radiographical, obvious or apparent disease. Once that happens, that's truly what we would call stage 4 metastatic disease, and that's usually not curable. I'm using the word usually now there very intentionally, because nowadays there are some patients, again, these are usually HER2 positive metastatic breast cancer patients that can sometimes be cured of their disease. Clearly not always. We've had some male breast cancer colleagues on this forum who have actually been HER2 positive and did well for years, but ultimately did succumb to the disease. What that also tells us, though, is many metastatic breast cancer patients can still be treated chronically for years, sometimes decades. And so the thinking nowadays is if we can't cure it, maybe for many patients, we can turn this into a chronic disease so that patients can live out their natural lives, similar to what insulin has really done for diabetes. All right, for those of you paying close attention, you might have heard me say that we can eradicate micrometastatic disease, but not true clinical radiographic apparent metastasis. And why is that? Well, once you get to a clinical apparent metastasis, something you can do on find on clinical exam or scans, that's already a billion cancer cells. And the problem with cancer is that it's a DNA disease, meaning that the DNA of a cancer cell is distinctly different from a normal cell. And there's also the problem of what we call genetic instability. That means every time a cancer cell divides, the two daughter cells are different at the DNA level. That's not the case with normal cells. In normal cells, when a normal cell divides, the two daughter cells are identical sibs. And so there's 100% or close to 100% fidelity. In cancer, that goes awry. And what that does is it leads to this scenario where you're constantly breeding what we call subclones or in tumor heterogeneity. The problem with metastatic disease is that, let's say it's here where it's a billion cells, then you throw any drug you have at, there's going to be pre-existing clones that go through what we call a bottleneck. And then that just begets even more clones and more drug resistance. On the other hand, in early stage disease, once we've taken out the primary tumor, and if there's micrometastatic disease, let's say it's on this level, then there's not that much heterogeneity. And so giving drugs like chemotherapy and endocrine therapy can potentially eradicate these guys, and then we can cure patients. So that's what I always have a conversation with when the first time I meet a new patient is, what are the goals of therapy? And in this case, systemic therapy, because that's what I do as a medical oncologist. Metastatic disease, as I mentioned, is prolonging life and treating symptoms. Adjuvant therapy is what I do in the curative paradigm. So it's adjuvant or secondary to the primary modality of surgery. And again, we're trying to eliminate micrometastatic disease based upon the rationale that I just showed. And to give you some scope of this, every year in the United States alone, there's over 150,000 patients who are eligible for these additional adjuvant systemic therapies. Now, you may be asking, rightly so, if you can't see the disease, if it's micrometastatic and microscopic, how do you know it's there? And how do you know that the therapies you're doing even work? And it really comes from large, randomized, multi-institutional trials that take years to decades to complete. And the studies are kind of like this. This is one of the early ones published in Lancet in 2005 as a review, which showed if you have two groups of patients who are very closely matched for their stage, age, type of breast cancers, and one group gets, let's say, placebo after surgery, then at 5 years in this example, 38.8% are going to recur. On the other hand, if you give that same group or the experimental arm polychemotherapy, meaning more than one chemotherapy, then you reduce that recurrence rate by about 13%. And that's shown here in this slide or this box, where in this particular example, surgery alone cures about 63% of breast cancer patients, but a third are going to die from their cancer eventually. With chemotherapy, we cure another 13.6% of patients. And though that doesn't sound like a lot, in fact, again, that's tens of thousands of additional lives that are cured because of what we do. That does come at a cost. Every year we know that there's going to be a small percentage of patients that will die due to complications of chemotherapy. And that's shown here in the blue bar. You can see that this is slightly bigger than the no additional therapy. But overall, we are doing far more good than harm. The hardest part about this paradigm is that we don't really know who's cured, who has microscopic disease, and that's something that we're working on with liquid biopsies, which I'll mention later in the talk. On the other hand, metastatic disease, as I mentioned, is largely thought to be not curable. We can use a bunch of different therapies now for hormone responsive disease, the so-called, again, ER/PR positive disease. We can use endocrine therapies, including tamoxifen or aromatase inhibitors. And we can also use other types of hormone therapies that are a little bit more old school by today's standards. The newer kids on the block with hormone therapies are something called CDK4/6 inhibitors. Some of you may have even been on this or know about these, but palbociclib, ribociclib, whoops, that was a misspell. That should be abemaciclib, ABEMA. And these are almost always given with hormone therapy, with the exception of abemaciclib. And then there's other drugs. Exemestane is one of these aromatase inhibitors, but everolimus is what we call an mTOR inhibitor. So these are all non-chemotherapy options for hormone responsive disease. For HER2 positive disease, we've got a lot of drugs. And again, this is why some of these patients are potentially curable. And Herceptin was kind of the first drug. Then there was Perjeta. We have Kadcyla, lapatinib, neratinib, tucatinib. Now we even have another drug called Enhertu, which I mentioned at the end here. These can be given with or without chemotherapies, with or without hormone therapies. Neratinib and tucatinib are also oral. And then for triple negative breast cancer, for the first time, we also have, I didn't put it on the slide, we have an immunotherapy. Oh, I did put it as well as immunotherapy. We have immunotherapy for a subgroup, but we also have this drug that's called sacituzumab. This is what we call an antibody drug conjugate. It's similar to this drug Kadcyla in the sense that it has chemotherapy linked to the antibody that directs it to the cancer cell. And so nowadays for metastatic disease, we really have to individualize choice. It depends on the receptor status. And the very newest kid on the block is this drug called Enhertu. This is probably the most closely related to Kadcyla. Kadcyla and Enhertu really are Herceptin, but both of them have chemotherapy molecules. The difference is Enhertu has about 7 to 8 chemotherapy molecules that's different from Kadcyla. And so it has a much bigger what we call payload. And the way these things work, they're not really targeting HER2 per se. They're actually just using HER2 as a docking protein. And then it allows them to get inside the cancer cell and it gets activated, releasing the chemotherapy and blowing it up inside and out. And so that also has what we call bystander effects, because once the cells are blown up, the chemotherapy molecules hang out for a little while and then will kill the neighboring cancer cells. Unfortunately, they also will have some side effects. And so Enhertu actually, because it has so many chemotherapy molecules, does have a higher toxicity profile than Kadcyla does. One of the things that I'm going to try to point out is that when you think about it, breast cancer was one of the first cancers that had targeted therapies because of HER2 and the antibody therapies that I just talked about, as well as now some of the other drugs for HER2 positive disease. If you even think of a step further back, estrogen receptor really is also a targeted therapy. It's not as precise because, again, there's lots of other tissues that have estrogen receptor, but clearly it's an important drug for hormone receptor positive disease. I'm sorry, important target for hormone receptor positive disease. So that gets to genetics and what I like to call precision oncology, which means different things to different people. But if I propose to you that all of these studies, these large randomized clinical trials led to, oh, this is what we do for stage 2 ER positive, HER2 positive breast cancer. That's how we treat everything pretty much across the board, very similarly, if not identically. But we now know that just like people are different, so are their cancers. Because as I said before, people have different DNA and therefore their cancers will have different DNA. And cancer is a disease of DNA gone bad. So what I like to think of precision oncology is really the genetics and the genomics of how we treat every patient's cancer in a very tailored fashion, or trying to make their healthcare personal. And we do that by sequencing the DNA. So just as a kind of background, cancer is like everything else. It's composed of bad cells, but in this case, cells are bad. But why are the cells bad? The cells are bad because their DNA is bad. DNA is the blueprint for all of our cells and therefore the blueprint of who we are. And DNA serves as a template for something called RNA, which itself serves as a template for actual proteins. And proteins are what makes us our cells, and cells are what makes our tissues and who we are. So you can imagine that if you have a mistake here, it will actually turn into a mistake here and turn into a mistake in a protein. And that's the problem with cancer. It's always changing and evolving. It's really Darwinian evolution, because you can imagine if these mistakes happen randomly, then many of them are going to be neutral, some of them are going to be deleterious to the cell, and some of them are going to be advantageous for the cell to live. But you have to remember the living advantage are the clones that get selected for. Again, strongest survive, and that's Darwinian evolution. So all it takes is one cell to develop a mutation that gives it a growth advantage, and it'll start to become the predominant population, and then it will beget even stronger clones because of, again, mutations and clonal heterogeneity. So about 15, 20 years ago, there was something called next-gen sequencing. First-gen sequencing was expensive, cumbersome, and slow. Next-gen sequencing kind of works like this. You just chop up a sample of DNA. So you take cells, grind them up, extract the DNA and the RNA, and then you make what we call libraries. But all they are little short fragments of DNA that you can now put on this machine, and it will sequence all the letters of the alphabet for the DNA for that sample. The key to this, though, it does all of these little fragments massively in parallel. So rather than the old way where we just sequence it serially, we'd have to just keep going right to left, right to left. This now just stacks everything together. And now that we have a reference genome sequence, which is the consensus of now hundreds of thousands, we align, the computer aligns all of these little fragments against the reference sequence. And it will say what's different about this sample of DNA compared to the reference sequence, and that's highlighted here. That's how we actually sequence and find mutations in cancer DNA, as well as inherited DNA. So this can sequence literally billions of base pairs of DNA. So each one of us, our DNA genome, if you will, is 6 billion base pairs, 3 billion from mom, 3 billion from dad. And so you needed some technology like this that could do this rapidly or massively in parallel, all those little fragments. We need to do multiple sequencing, because if you were just to sequence 6 billion base pairs, you wouldn't actually get everything because it's somewhat random. And so going back to this analogy, some of your fragments will stack at the same area, others will only stack a little bit here. So the more you do, we typically say nowadays 10 to 100-fold coverage, meaning that you have to do, instead of 6 billion, you have to do 60 or 600 billion base pairs of sequencing to make sure that everything is sequenced at least once. The cool thing about this technology, which you also couldn't do with first gen, is that you can get down to what we call allele frequency. So even if your mutation, like here, you're seeing it represented in about half or more of these DNA fragments. But let's say in cancer, there was only one. You can now do 1 to 100, 1 to 1,000. Sometimes you can do 1 to 10,000, and you can still detect that. And that becomes important for what we do in the liquid biopsy space, as I'll mention. The other thing is you don't have to do everything, the 6 billion base pairs. And we rarely do, actually, because most of that stuff is not helpful for cancer. So we actually pare it down and we just rip away gene fragments for the ones that we're interested in sequencing. And so that dramatically reduces cost and time to get the sequencing results back. All right, as I mentioned, cancer is a disease of DNA gone bad, but it's not just one genetic mutation, it's the accumulation of multiple genetic aberrations or hits. And so if you look at these green cells as normal cells, this is the way cancer happens. You get one mutation, that by itself isn't enough, and those cells will proliferate because there's a selective advantage, again, Darwinian evolution. And then in that population, you get another one, and that becomes the predominant clone because it has an additional growth advantage, and so on and so forth until you get cancer. And it's estimated you need about 3 to 8 mutations to go all the way from the green to the red. The other thing that can accelerate this process is if you're a BRCA1 or BRCA2 carrier. And that's because you've inherited that first step from either mom or dad. And that's shown here in this diagram. I'm not going to get into the weeds of this, of what tumor suppressors are, but you can see from what we call these genetic pedigrees, this is an example of a different tumor suppressor gene called retinoblastoma or RB1. And this circles represent females, squares represent males, and the colored ones represent people who have the disease. And in this case, these are tumors of the eye. You can see here, if it's inherited, this means that dad has one bad copy, one good copy, and then passes on to their son, who inherited, unfortunately, the bad copy instead of the good copy. So that son is going to get the disease. It still requires additional genetic hits, and that's shown in here. On the other hand, you can also get non-inherited forms of retinoblastoma, and that happens here, where everything looks the same and normal, and then you get one hit later on in life, and then in that tumor cell, you get additional hits or genetic mutations. So this accelerates the process by about a decade. We think that there's about a mutation per decade, and that's how we can get cancers, when why most cancers are about usually in the 5th, 6th, and 7th decade of life. Not always, obviously, but if you inherit one, that gives you a leg up, so to speak, to get cancer. And so you've knocked off maybe about 10, 15 years. And that's why typically in patients with BRCA1 and 2, they'll get breast cancers at a younger age. All right, so the inherited genes, we call that germline, and the testing for that is germline. Again, every male breast cancer patient should be tested for a panel of genes now, but especially BRCA1 and BRCA2, and especially BRCA2. The testing has evolved over time. As I just mentioned, we're now using next-gen sequencing instead of the old standards. And the panel gene testing, as I told you before, has become routine. So we're not testing, there's 22,000 genes in our genome. We're not testing all 22,000, we're testing at most about 100. And that's because the majority of genes that we know about are occurring in those 50 to 100 genes, with actually the most majority being in BRCA2 for male breast cancers. So, but who's to say that in a family that has a lot of cancers and all these testing gets negative, that it's just one of the other 22,900 or 21,900 genes. So that's what I always want to make sure is that people who get these tests need to also see genetic counselors, understand what the tests can show and what they cannot show, and not to be fooled into thinking, oh, just because my gene testing was negative, despite having a strong family history, that means I'm okay. That does not mean it's okay. We have to presume that that patient probably has something else that we're just not testing for in 2022. The other thing that's really changed over the last 5 years, before I would tell patients, this doesn't have any bearing on your therapy. It's really about knowing who's positive and testing your blood relatives. That has definitely changed. Now we have drugs against BRCA related, 1 and 2 related cancers, not just breast cancers, but ovarian cancers, pancreatic, prostate cancers, et cetera. And they're called PARP inhibitors. These are already approved, no longer in clinical trials. So if you have one of these mutations, it now has therapeutic consequences. Similarly, there are some families, they typically can get colon cancer or endometrial ovarian cancers called Lynch syndrome patients, and they have inherited gene mutations in what we call mismatch repair genes. And it turns out if you have one of these types of cancers, you can have dramatic effects against immunotherapies or what we call anti-PD-1 and anti-PD-L1 therapies. So again, genetic testing, germline testing now has true therapeutic actionability. All right, this is tumor-only testing now. I'm changing gears. So this is when we take out a tumor, we grind it up, and then do the next-gen sequencing. And in the old days, we only did maybe 50 genes, and now it's expanded to several hundred genes. And actually, we may probably be seeing on the horizon what we call whole exome sequencing, meaning that you actually look at all 22,000 genes, but you're just looking at the coding regions. So whether or not that's really going to prove useful, time will tell. The other problem with that is the bigger the panel of genes, the longer it takes to sequence. It can be more expensive, although that's coming down. But the hard part is you get a lot of noise. Everyone's DNA is different, as I said earlier. And so then it becomes a problem. Despite having a reference genome that's a summation of 100,000, hundreds of thousands of people now, you have to remember we have billions of people in this planet, not trillions. And so that's the hard part. When we see something, sometimes we get something called variants of unknown significance, meaning we have no idea whether this is really related to the cancer and causing cancer, or is it just like who the normal person is. And when we do tumor-only testing, we don't have to consent. We do have to consent for germline testing or heritable gene mutation. All right, the reason why this has become important is because as the panels grow, as we get more confusion, it turns out when you think about it, the best way to know whether or not in the tumor a mutation is important or not is just to compare it against its own normal counterparts. That's what we call germline filtering. So what we do at Vanderbilt now is we take a tumor, and if we can, we get blood. We sequence both, and we compare the two, and we say, aha, this is the stuff that's only in the tumor, which is a higher likelihood to mean that it's probably important, as opposed to if it's just in the normal cells as well. And that's really an advantage. There's a potential disadvantage because some companies early on were filtering out because they would say, okay, here's a normal, here's the tumor. We're just going to subtract everything that's in both because that's just the heritable stuff that we don't care about. But if you didn't know you had a BRCA1 or BRCA2 mutation and you had one, that would also get filtered out. And that's done at the computer level. There's no human doing this. So there was missed opportunities. And so now many companies, including the one we work with, Tempus, they will actually tell us, hey, you know what? There's a BRCA1 mutation here. And they have a list of genes that's recommended by the American College of Medical Genetics and Genomics that say, if you find these things, you should actually report them out. This gets a little bit more complicated if it wasn't already, because you have to understand how this works. And many doctors aren't trained in this. So this is one of my favorite examples. This was a test where we had this is from a molecular tumor board, which I'll explain in a second. It was a head and neck cancer from the right buccal mucosa. So that's the inside of the mouth. And it was a very high sample. It was mostly tumor cells, 75%. But look at the source of normal DNA. You can use saliva sometimes or blood just to do that filtering that I'm talking about. Well, you got to remember though, saliva for a head and neck tumor is going to contain tumor cells. So that's not really a good source of DNA for normal filtering. And these are just some of the subtle things that get lost when you're doing these tests. This one came out blurry, but I like it because it's also what I call unclear. Many companies, if not all of them, you don't have to send the source of normal DNA. And in this particular case that we discussed, there was no source of normal DNA. And so when you interpret the results, you have to take that into account because it's showing everything. And in this particular case, I don't think I put it on the next example. I didn't. There was an androgen receptor variant, and the prostate cancer doc came up to me and said, "Hey, doesn't this mean there's an androgen receptor mutation?" And it turns out you look at it, and I could tell, well, there's no normal DNA. This is just a normal variant of the patient that they inherited from mom and dad. It has no bearing on the cancer. Again, these are some of the subtle tricks that we have to watch out for. This is a case that shows and highlights two other things in breast cancer, including male breast cancer, that we can now do drugs for. So there's something called high tumor mutation burden, and this is one of the first what we would call tissue agnostic indications for immunotherapy. What this means is that if your cancer cell has tons and tons of mistakes, there's a higher likelihood that they're going to respond to immunotherapy. And this was a patient that we treated. The threshold is 10. This patient had 36.7 and had a very good response to immunotherapy. I liken this to the analogy of playing the lottery. If you buy one lottery ticket, that probably doesn't really enhance your odds of winning. But there's a certain threshold, if you had enough money, that if you bought, let's say, 1 million lottery tickets, your chances go way up. Now, you've also spent more money, but that all being said, your chances go way up that you're going to win the lottery. That's what tumor mutation is, because what we're really measuring is how different is the cancer cell from the normal cell. And that's what the immune system really needs to attack cancer. It's much easier for the immune system to recognize different cells than normal cells. And that's why tumor mutation burden has become this marker. So literally any solid tumor, male breast cancer, female breast cancer, prostate cancer, if they have a high tumor mutation burden, they can actually get immunotherapy now. The other thing that's been really interesting in our field is looking at what we call gene fusions. This is what happens when, instead of mutations, you get one gene here and another gene here, and they break pieces off and they inappropriately get stitched together. And that's what we call a fusion or a rearrangement. Turns out there are now really good drugs, and some of you would see me present this case of this little girl in Bangladesh. This is very similar to that. It's the same fusion of the genes called NTRK, or they're NTRK 1, 2, and 3. And this particular patient, it's a breast cancer, triple negative, had a rare, these are really rare, like less than 1%. But if you have this, we have now 2 FDA-approved drugs that can give you remarkable results. And so this is something that we're looking for. And we even have the database here at Vanderbilt that we can go back and find patients that we had sequenced and find these, what we call fusions or chromosomal rearrangements. So this is another really, again, great tissue agnostic indication. Any solid tumor that has any of these NTRK fusion genes, you can get one of these 2 drugs. Now, I mentioned the Vanderbilt Ingram Cancer Center. One of the strengths here is what we call informatics and health informatics, as well as bioinformatics. But that allows us, we have this enormous database now. And this is part of the reason I came here, that we can actually find our patients. And even though we don't have drugs for them today, once something comes out, we can go back. And that's what we did for these high tumor mutation burden patients. We went back to our database and said, hey, how many of these patients, however rare, have we sequenced before that we know they're still alive? And then we can contact their providers and the patients themselves and say, you know what, you've got this thing and a new FDA drug was approved. That's hugely important because it allows us to shorten that gap between knowledge that we have today being disseminated out globally. This is another area of that. This is a study I did, probably the last one's when I was at Johns Hopkins, where we just decided, you know what, let's just sequence every metastatic breast cancer patient and see if we can find inherited mutations. And sure enough, we found way more than we were anticipating or what our guidelines say. Subsequent to this, the guidelines have been updated, and I'd like to think that we contributed to that thinking. But it turns out you can really find a lot more inherited mutations, and we've been doing that in the effort here since I've been to Vanderbilt. One of the scary things to me, though, is that 2 of these patients were two of the BRCA2 patients that we found were previously recommended to have testing, but they didn't get it done for whatever reason. And as I just told you, we now have really great drugs against these types of cancers. So this is a huge problem, and why I'm committed to doing talks like this so you all can spread the word. You need to get tested because it's a therapeutic consequence. Now, finding these fusions is not so easy. And so you also need to make sure that if you get testing, you get the right testing. In the old days, it was just based upon DNA, but it turns out we can use RNA to find them now. And I told you that early diagram, how DNA turns to RNA turns to protein. Well, when you get DNA to RNA, something else happens. You get rid of these black lines that we call introns. The way next-gen sequencing works is that you have to do a lot of sequencing. You may miss fusions because it would just be by chance that you have something that crosses where one gene is inappropriately stitched together with the other one. On the other hand, your chances are much higher finding this if you look at RNA because you've gotten rid of all those black lines. And so again, if these are all these little sequencing reads that I showed you before, it's much more likely that any one of them is going to hit this junction and say, hey, you know what? You have two genes that shouldn't belong together on here, as opposed to here, where you may or may not find it depending on how many reads are in that area. So that's really, really important because right now, only one or two companies are doing this, where we're doing all the RNA and we're looking at all the different fusion transcripts that can occur. And I will tell you, it's not that common, but we have found a number of novel fusions that would never have been found by any other methods. So again, and some of them are really of therapeutic consequences, like these NTRK fusions. So this is something, again, make sure that even if you do get the testing, you really get the testing that leaves no stone unturned. This is a couple examples of this. This is not in breast cancer, but just to highlight the point, this is in non-small cell lung cancer. There's a 43-year-old male who had brain mets and went on trial, and we ordered, our vendors called Tempest, and we discussed it. So the first thing he had was this, what we call rearrangement or fusion, and there's a really great drug against ROS1 fusions. So he ended up getting that. He did really, really well, but he eventually did progress. And then we did repeat testing, and then we were surprised. We found, again, only by RNA, this fusion. Now, it turns out this fusion, this gene, especially RET, is a mediator of drug resistance against the drug that was being used against the original target. But now, literally just in the last 2 years, we have a really great drug against RET. And so that patient now is on this and has responded. So again, it's one of those amazing success stories that if you sequence, you can find things, and we have drugs for some of these things. And yet this is still not being done enough or as widespread as it should be. I think the last kind of example for the sequencing that I want to talk about is RNA, not for the tumor, which is what I was saying before with the fusions, but looking for new mutations in genes like BRCA1 and BRCA2 that traditional DNA testing will not find. And so when we look at BRCA1, BRCA2, we are only sequencing, going back to this example, these coding regions or these boxes. And that's because the black lines, the so-called intron sequences, are really huge between the coding regions. It would take extra time, extra money, etc. And so we actually have just ignored the black line regions for the better part of 2 decades in terms of commercial testing. It turns out occasionally you can have a mutation in a black line that affects how that gene is being expressed. It will terminate it and make that gene functionally mutant. And so the best way to do that is to first look with RNA. And so we all have 2 copies of every gene, except for X and Y if you're a male. And so nowadays with RNA, we can do the sequencing and see whether or not there's what we call imbalance. And that's kind of shown here. Let's say this is the gene copy you inherited from dad, and that's the little blue dot, and then the darker blue dot is the gene copy that you inherit from mom. Because everyone's DNA is different, including mom and dad, we have these things called SNPs, or single nucleotide polymorphism. So we can distinguish between the gene copy from mom and the gene copy from dad at both the DNA and the RNA level. What that tells us, when we see an imbalance, that means, hey, something's wrong. In this case, with the little blue dot, the one we inherited from dad, we're not seeing as much DNA or seeing as much RNA from that gene copy as compared to mom. That's a smoking gun. And then if we see that at the RNA level, the companies will then go back and sequence these black lines. And it's not very common. It's like 1 in 1,500 right now, but you can actually find these. And this is actually an example of a man with prostate cancer. And he actually had what we called a VUS in the BRCA2 gene, got RNA testing done and showed and proved that, in fact, that's not a VUS. That's a real pathogenic mutation, because what it does is it makes that RNA not form. And that's the, again, what mutations do. They take away the function of the gene for BRCA1 and BRCA2. The implications of this, as you can guess, are tremendous. This is a guy who had metastatic prostate cancer. Now we can get these PARP inhibitors, the therapies for BRCA1 and BRCA2, which are highly effective. Moreover, his family members can now be tested for this exact same BRCA2 mutation. So again, testing. That's really the key. Liquid biopsies, as I mentioned earlier, is something that we have a lot of expertise within my lab group and now at this institution. This is relatively new, but now it's been around for almost a decade. And it's this whole idea, I'll go to the next slide, that all of our cells secrete free-floating naked DNA into the bloodstream. We don't know if this has any functions. Many have heard me say I call it cellular poop because we just think it's a waste product. But if you look here, all the normal green cells are shedding normal green DNA, but also cancer cells will shed their multicolored DNA, and that represents mutations, and also in this case, these fusions with these big X's. So this is a good biomarker because the plasma life, half-life of DNA, the half-life of plasma DNA is very short. And then unlike what this drawing shows, however, usually you only find one mutant molecule in 1,000 or 10,000 in a tube of blood. It's overrepresented here in this, and it is the proverbial needle in the haystack. But as I mentioned earlier, with next-gen sequencing, that's okay. We now have the capacity to detect one mutant molecule in 10,000. So we're now testing whether or not we can use these tests to detect patients who have microscopic disease, patients who may be cured, but we don't know if they're cured. And I think that's the real exciting part in the liquid biopsy space is, can we use these blood tests to someday say, hey, you're cured, you don't need to do additional therapies, or you're not cured, and we should continue to do more therapies to try to cure you. This is really, again, still Buck Rogers science fiction, but it's really on the horizon and very exciting. All right, so there's other things that we have to be careful for liquid biopsies, just like tissue biopsies, and I'm gonna go through this case very quickly. This is a 70-year-old with metastatic breast cancer, was originally diagnosed in 1998. Strong family history of breast cancer, but had never had genetic testing, again, germline testing done, did not have anything that was easily biopsyable. So a liquid biopsy was obtained. And here are the results. This is a very common mutation that our lab helped discover way back in 2004 called PIK3CA. There is a drug for this now if you have one of these mutations. And back then there wasn't, so don't believe this. This is completely wrong by today's standards. There's also a BRCA1 mutation that was found, but it was on a very low frequency. This told us that this probably was not inherited, but probably was what we call somatic or sporadic. So it's just in the cancer cell. At the same time, these can still respond to PARP inhibitors. These are all the different PARP inhibitors, and there's more now. And then there's also a TP53 mutation, which isn't unfortunately druggable. These frequencies are really low, and that gets back to what I was saying before. You're really measuring cancer DNA as the numerator against the background denominator of normal DNA, and that's why they're so low. If you were to take pure cancer tissue by comparison, these would usually be in the 30 to 50% range. All right, so our case report was that, we weren't sure, but we thought that it was probably, this was not inherited because it was so low fraction. But at the time, we'd still recommend it because that patient really needed to get it, but was in fact negative for BRCA1. And so our initial thought was there that we would actually, wow, this is ancient. It shows you how the field has changed. Back then, we did not have the data we have now that PARP inhibitors would work against tumor-only BRCA1 mutations. We actually do have that data now. So in 2022, I'll have to update this. I just realized it's now outdated. We would recommend this. The other thing is that we didn't have a Pellicib, so we recommended a PI3 kinase inhibitor trial, but we now do have a drug for PIK3CA mutations. All right, I'm not going to read all this, but I think you probably get a sense that this is complicated stuff. It's one thing to get the data, it's another thing to interpret. And these are all the questions that have arisen over the years because of this genetic sequencing. So back in I don't remember anymore, I started with the first molecular tumor board at Johns Hopkins, and might have even been the first one in the country. And this is some of the publications that came out of it, and some of the makeup of what we did in terms of who was on the molecular tumor board, the difference of expertise. We had patient advocates from the very beginning, which we still do. And then when I came to Vanderbilt, this is, again, I'm not going to belabor these points. I started a very similar type of tumor board, and we called it the Big Hope Tumor Board for Hereditary Oncology Personalized Evaluation. And who doesn't like the acronym HOPE? But the other thing that really occurred to me is something I got involved with the Global Cancer Institute. I think you heard me say earlier, part of the problem now we have when we're getting a new diagnostic and a new therapy, we're almost a victim of our own success. We create instant health disparities because only big tertiary academic medical centers can offer it, and only those who are well-resourced and well-knowledge as patients will actually refer so their patients can get it. And it can take up to a decade before there's widespread adoption. And you think about how many patients could be helped in that interim, just simply don't know, and their doctors don't know. So I was part of something called the Global Cancer Institute since its inception. It was founded by Dr. Paul Goss at Massachusetts General Hospital. It was originally started as an Avon Centers of Excellence, and we brought together all sorts of breast expertise, breast cancer expertise, and we try to help low middle income countries around the world with their breast cancer cases. So these were the summation of the sites that were involved at the time. I phenocopied this with molecular tumor boards. And so through that Avon network, I have a lot of colleagues throughout the world who now have access to these genetic and genomic tests, but they don't have expertise in how to interpret it. So we meet once a month. It's called the International Virtual Molecular Tumor Board, or I call it Our Cancer Genomes, which is a play on words for a database here called My Cancer Genomes. And I need to update this. We're probably close to 50 sites, a mixture of academic, community, and international sites. And Paul actually has stepped down. He asked myself and Don Dizon, who's my partner in crime, to take over. So we are now leading the charge of getting genetic testing for every country in the world, if we possibly can, for patients with breast, ovarian, and other types of cancers. So with that in mind, hopefully I'll convince you all, and we'll have some time for questions, that gene testing for all breast cancer should be routine standard of care, should include the tumor in patients with metastatic disease, and germline, again, hereditary cancer DNA for all men with breast cancer at any stage. Our next steps, I think, are to democratize or disseminate and implement this to all patients across the state, country, and world. And I'd like to say, even though I'm obviously biased, that Vanderbilt Ingram Cancer Center is really leading this efforts with partnerships, and I hope we are really, I know we are, at least the patients I've seen here, changing the lives of patients with cancer. Tons of people to thank, but in the interest of time, I'm going to stop here and try to take any questions, and thank you for your attention.
Bravo, as usual. Ben, this, for anyone who doesn't know on this call, Ben is actually one of our medical advisory members, and he is our go-to man whenever we have a question, or one of you has a question about your treatment or your care, or you send reports. We connect you with Ben, and he answers immediately, except on the weekends. We give him weekends off for his family. And we cannot thank you enough.
No, my pleasure. And, you know, I saw your chat message to me. I would love to talk further, maybe offline, here, about how GCI and the Alliance, Male Breast Cancer Global Alliance, can actually work together. I think that would be fantastic.
I think it'd be great. Yep. Questions, anybody? Michael.
Thank you, Dr. Ben Park. Again, outstanding presentation as usual. When you're talking about detection of cancer DNA in the plasma, and I'm just talking early stage patients, is that done before or after surgery, or both?
Early stage, we're not doing that yet outside of research. There are some people who are, because there are tests available for colon cancer. But, you know, this is one of the areas where I'm a little bit cautious, because even though so colon cancer has what we call validation data, meaning that companies have taken clinical research and shown that if you're positive for liquid biopsy, this is after surgery, okay, and you've done all the standard of care, then you probably do in fact have disease that's going to recur. What they don't have yet is what we call negative predictive value. So what I talked about earlier was positive predictive value, right? If you're positive for ctDNA or liquid biopsy, you're probably eventually going to develop metastatic disease. And then we don't have negative predictive value, meaning that if you're negative, that doesn't mean you're cured. We haven't created the technology that's deep and sensitive yet. I think we're going to get there, but we're not there yet. But what I'm really concerned about, Michael, is that we don't have utility data. We have validation, but not utility. And what that means is, what do you do with that finding afterwards? You know, it's one thing to say, well, it sucks for you, you're going to get metastatic disease, or maybe you already have it. It's another thing to say, well, let's put you on a drug and show and prove that that's going to cure you.
Yeah.
We're starting to do those studies right now, but I feel like the companies and others have jumped a little bit too far ahead. This just happened with a patient that I know a colleague had sent this test and they were positive, and the patient and their family were rightly freaking out. And, you know, from the one hand, the oncologist side was like, well, this is good because if you have metastasis, we'll catch it early and we can try to treat it earlier. But I said, you know, I bet you based upon how low that frequency is, that amount of ctDNA, that the scan's going to be negative. And sure enough, it's negative. So now what do? Nobody knows. Do you just keep drawing more blood? You keep doing more scans, exposing patients to more radiation? We have no clue. And so that's what I've been trying to tell companies whenever will listen. It's amazing. Sometimes the deaf ears were just like, no, but. It's like, and then I say, no, time out. The real but is we don't have any data that this helps patients. And that's why it's also not being covered by insurance for the most part, because there's no utility that has been proven with this. I do think it will eventually. I mean, again, my lab is heavily vested in this, but I often quote Dan Hayes, who is the breast cancer guru at Michigan, program leader. He's always saying, and he does a lot of biomarker development, a bad test can be just as dangerous as a bad drug. And I think that's really true. We need to make sure that these tests are as good as we think they are by doing the rigorous science, and that just takes time. So to make your short question answered long, that's why we're not doing it yet routinely for early stage.
Okay. And one follow-up question is, in light of the pandemic, has Vanderbilt ramped up to 100% as far as your labs and everything, getting back to business?
Yes, although there's been some caveats that, as you can well imagine. So, you know, it's getting better now, but if people end up being COVID positive, they have to go out for 5 to 10 days, depending on when symptoms start, et cetera. So that has created in some circumstances, we're okay right now, but boy, back in like January, February, March, it was like a rotating door of personnel going in and out. So we actually, in January, we were already short-staffed because of the Great Resignation, but because of everyone getting Omicron variants and having to quarantine, we had to shut down our clinical trials here for 3 weeks.
Wow.
And that was really, really painful, but we just couldn't safely put on patients, and we had to do the right thing. We're actually not as worse off at some of our sister institutions where they had to shut down trials for months. And again, not because of the initial wave of the pandemic, but because of all this stuff that's been happening.
Wow. Well, thank you, and congratulations on all your advancements.
Well, thanks, Michael.
Matthew, did you have a question?
I did. Thank you. Hi, Dr. Park. This is just fascinating to me. And it's
Thanks. And please, I go by Ben or Dr. Ben, okay?
okay, Dr. Ben. Thank you. This has really been incredibly helpful. The whole day has. So thank you for this, Sherry, and everyone. But I'm actually in a trial for the ctDNA testing.
Oh, good.
Yeah. So, and it's really helpful actually to hear. I mean, it helps me to contextualize, you know, how to view these results that I'm getting, which are always negative, by the way. So, you know, it's good. That's hopeful, I guess.
Yeah, it is.
But yeah, I'm just fascinated to hear this. And I don't really have any questions. Honestly, my doctor has been very upfront with me about what all of it means. And she's been very good about communicating results and that kind of thing to me. But I have to say that hearing you, and there was another speaker earlier, talk about, you know, what do you do with these results if all of a sudden it should come up positive?
Yeah.
It's hearing this actually helps me to put things in perspective so that I'm not pinning all my, hopes and dreams and future health on.
Well, I'm hoping you're going to be part of a study that does have a pathway to get on some sort of therapy, whether it's experimental therapy or existing therapy, because that's where the field's headed right now. Now that we know we can detect this, and that means usually for higher risk, think of that almost as a safety net, that if it's positive, get on a trial now to see whether or not we can clear that. Presumably they would scan you first, but likely, again, it might be negative, and then see whether or not we can eradicate it. When I was first starting this business, there was great hesitancy because people would say, well, how do you know that means anything if they're ctDNA positive? And I kind of felt like beating my head against the wall. Like, are you kidding me? This is cancer DNA. Of course, that's not going to be good. There are some caveats to that because there's some artifacts that we call like clonal hematopoiesis, et cetera. But for the most part, we know what the mutations in a breast cancer are going to look like. And if you see that, that's just nuts to me that you wouldn't act upon it. But it's taken this long to convince colleagues across the world that, hey, you know what? Now we know if you have cancer DNA in your blood, that's not a good thing. So study after study has now confirmed that that's a bad prognostic factor because it does mean the cancer's still there. Kind of no duh to me, but but that now has led people to really start gravitating towards, okay, now that we know for sure, as much as they can be competent, we will see whether or not we can treat patients, and that affords better outcomes. So that's really the utility studies that I'm really looking forward to helping, and we're doing some of it, but colleagues across the country and the world are now too.